pure materials, how to do the non-structure related impurity content in organic pure materials. We are going to discuss this topic. We have with us Dr. Arvind Gautam. He is the Senior Principal Scientist at National Physical Laboratory, New Delhi. We have with us Shri B.N. Dixit, our Honorable Director, Sir, Director Legal Metrology. Professor Gautam is currently working in the capacity of principal scientist. He is a graduate and postgraduate in chemistry from Chaudhry Singh University, MTech and other qualification from Punjab University. He has total 90 years of experience in the area of petroleum, polymer and chemistry. He has total 40 national and international research papers and one patent. He is technical assessor for NABL ISO IC 17025 and former member of ISO Working Group 18. Has supervised 10 number of postgraduates and PhD students. He has handled several external and industrial projects as principal investigator. Thank you very much, sir, for accepting our request and leading the talk on this very important topic, how the industry can get it benefited from your topic. We have with us Shri B.N. Dixit, our director, sir. Sir, would you like to speak 2 words, please?
Yes, Astoji.
Please, sir.
Sri Astoji, Director Legal Metrology, Government of India. Sri Arvind, Dr. Arvind Gautam, today the expert of the program. I think the Legal Metrology Act is sufficient for the measurement of quantities and other standards has been spread throughout the country. The today topic, the organic product, it is generated mainly from the original product, which are produced from the compost and other natural things. Natural water is also the organic product. Like that, there are so many trees are there which are growing and producing fruits and all that very things without any unorganic fertilizers. So those are also called the organic things. But the main highlight will be given by the Dr. Gautam Jha. So I request Director Legal Metrology to request Dr. Gautam to speak on the said topic. Thank you.
Thank you. Thank you very much, sir. Thank you for your all motivating words. We are actually looking towards you always for the development of these standards in the country. Thank you. Thank you very much, sir. Thank you. Over to you, Professor Gautam, please.
Okay, so good morning to one and all. At the outset, I would like to thank you, Mr. Asutosh Agarwal, sir, as well as special thanks to Mr. B.N. Dixit, former director, for inviting me for this special lecture series. And I also thanks to all the speakers presented in hybrid mode. So, good morning, all. Once again, myself, Arvind. I am a scientist in National Physical Laboratory. So today I will discuss about the non-structure related impurity content in organic pure material. So, I will cover what are the steps involved in the assessment of the impurity in a organic material in my presentation, and at the end of the presentation, if somebody have some kind of query or questions, I will try to answer at the end of this session. So first let me start with the introduction part. So what is the role of the purity in the organic compounds? So as I mentioned, purity is crucial for accurate characterization and reliable results in organic chemistry. So that is very much required, especially in the field of the pharmaceutical application, as impurity can lead the or interference with the data, which maybe have the harmful side effects. So When we talk about the reproducibility and interpretability, so purity assessment is essential for ensuring the reproducibility as well, because if impurities are presented in the organic compound, so it may mislead the research data, as well as accurate characterization is also affected by the impurity, because purity is the key factor when it comes to the true chemical composition of a substance. So accurate characterization is essential, but the only thing, if the compound is pure, then we can only do the accurate characterization. And if you're talking about the novel compound in research, if your compound is not purified, then your compound with biological activity cannot be taken into the consideration. So the simple question is, what is the purity of a organic compound? So as per the 1971 IPUSC organic purity monograph, the practical definition of purity is an organic material is sufficiently pure when the content of its primary component has been assigned. There are two components in a organic compound. One is called primary component, other is called the secondary component, which is termed as a impurity. So which may interfere with the purpose for which material is required. Suppose we require the material for organic material for the pharmaceutical applications. So whatever the primary component and whatever the secondary component, both component is important. So until unless we are identifying the impurity, we cannot do the or use it as a pharmaceutical applications. So every impurity, here we are trying to connect with our 7 SI unit. So one of the SI unit is mole. So mole related to the mass of a organic compound. So via gravimetric method, we can or for like organic compound uncertainty contribution associated with gravimetric operation and with the molar mass value are generally negligible relative to the uncertainty contributed to purity assignment. When we talking about the purity assignment, we are using the mass balance method. In next slide, I will explain what is the mass balance method. So impurity be term related to mole, realization of the mole. So with an example here, I try to explain what is the purity of organic compound. So as I already mentioned, purity is measure of the mass fractions of the primary organic component, which is called PC, present in the material. And it can be reported as percentage in the unit like kilogram per kilogram, or in submultiple of their like milligram per gram. So 90% purity means 0.99 kg by kg, or you can say like 990 mg per gram. So what is the advantage of taking milligram per gram? Because the main component and the significant impurity have the integral number at third place. So, what are the good practice to quantify secondary component, which is termed as impurities? Many secondary component are essential. So, what may be the secondary impurities like water of hydration, acid salt, and PC in mass fraction we can termed as weight kg by kg, readily converted into amount of substance of content like Kelvin mole per kg. So assigning of SI traceable value for the mass fraction content of the primary component of a high purity organic material achieve fit for purpose realization of the mole for the primary component. So, what is the method for purity assignment? There are four possible approaches I have mentioned here. So, like assign mass fraction W1, W2, and so on for each secondary component and subtract from the limit value. Like we have a 100 gram of the organic compound and we assign the mass fraction of our impurity like W1, W2, W3, and so on for each secondary component. If we subtract all the impurities from the 100 gram, we got the value of the primary component. So this is called the mass balance method for the purity assignment. This is the versatile method and widely used by the chemist. Another method is assigning mass fraction of the primary component by QNMR, quantitative NMR spectroscopy. As you know, quantitative NMR spectroscopy is the primary technique. So by QNMR, we can find the value of the organic purity of a organic compound. Another method is assign mass fraction of the primary component using another primary assay technique like colorimetry or acidometric titration. So this is the another method. Last method is assign directly total mass fraction uh, of all the secondary components subtract from the limited value. Like we can use the thermal method such as adiabatic calorimetry or freezing point depressor. But this is not a very popular method, and it's having a small measurement uncertainty but have the limited application. So here we discuss about the mass balance method for purity assignment. So four category of the secondary component impurities. One is related structure impurity, which is called SC. RS means related structure impurity. So absolute mass fraction is called WRS. Why we put WRS? Because this terminology will be using again and again in the some of the formulas. Or we can write SCRS, relative mass fraction of relative to primary component, by the chromatography. If we use the chromatography for calculating the secondary component, then we can write SCRREL like that. So water we can uh, calculate it as the secondary component, and W means water at mass fraction of WW. And other impurity may be the residual solvent and other contaminants, volatile organic compounds, which we can categorize as SCOS at the mass fraction WS. Some other impurity may be the non-volatile impurity, which we termed as SCNV at mass fraction WNV, with potential contribution from either or both, maybe inorganic impurities or maybe non-volatile organic impurities. So total non-related structure impurities, if we add them, and we also add the relative mass uh, mass fraction of relative to PC from the 100, we can get the primary component. In another formula, you can see if you minus uh, from 1000 to the relative W relative, and means this is non-related structure impurities. The formula is almost similar, but it give the more precise value. So how assay traceability will work in organic analysis? So we'll start from the first measurement procedure. So when we measure something, definitely we if we link it with 7 SI units, so our result are traceable, and it will be fit for purpose. So SI traceable purity assignment procedure, that is mass balance or QNMR, we are using for the certification of analyte mass fraction present in a pure material in mg per gram. So that is called the reference material. If we follow this procedure, means our measurement are site traceable, our purity is also site traceable, then we can taken this material as a reference material, and that is called the primary reference material. So realization of the SI unit for the analytes pure substance CRM, which we call mg per gram, and it's have the specific value which is mentioned on the uh, certified reference material certificate. So primary reference measurement procedure we can uh, uh, perform through the gravimetric method. Gravimetry is also a primary method, so which have the very less measurement uncertainty. And then it is going to primary calibrator, a solution of PRM, yani primary reference material, which can be used as a calibration solution of the CRM, and the concentration is taken in nanomole per liter, and that can be used in laboratory measurement procedure like HPLC, MSMS. So, like that, SI traceability in organic analysis can be established. So, method for the determination of the water content from the organic compound. As I already mentioned that impurity can be in the form of the water, in the form of the some other organic chemicals, or in the form of the inorganic chemicals. So first we'll discuss about the how to estimate the water content from a organic compound. So there are two versatile technique available. One is Carl-Fisher titration, another is thermogravimetric analysis. And apart from this method, if your water is present as a residual solvent content, which is called WOS, then we can use the GCMS or GC with FID, or QNMR or TGA with MS. If it is a non-volatile water content uh, or impurities, then we can use TGA, ion chromatography, and ICP-MS. But here I will actually explain only two techniques, because time is not permit me to explain all these techniques. So before that, I would like to tell you something about how to make the accurate mass measurement with SI traceability, because measurement is a very much essential part of this entire process. If you want to do the yeah, purity analysis of organic compound, so mass measurement is very much important. So what what are the procedure we are using here? As I already mentioned, gravimetric procedure will do like mass of powder and mass of solution, so we can get the value of mass fraction of content of standard solution. So uh, uh, SI traceability will link through the kilogram and then mole. Okay, so traceability to mole require SI traceable mass fraction assignment for the purity of the source material. So there is a great history of traceable mass measurement in the past. Like mass standard was also found in the Harappa civilization or Indus Valley, and in the Book of Dead we can see there is a balance they are using, and some of the balance in Virmir 1664 woman holding a balance, a steelyard kind of balance, and pen balance. Balance in the 21st century, ampere balance come into the picture, and now Finally, we at this is at this juncture we are using the Kibble balance, which is directly based working on the principle of the Planck constant, which is the fundamental constant. Now comes to the redefinition of the SI unit. So in 20 May 2019, four SI units are redefined. So the mole, the kilogram, the ampere. Uh, there are total four SI unit which is redefined, and they are directly linked to the fundamental constant. Some more analytical balance which is based on the electromagnetic force compensation, because when we weighing something, electromagnetic force can compensate this gravitational force, and this technique is more advanced, having the less measurement uncertainty. So I will talk about the gravimetry. Uh, what is gravimetry? What is the hypothesis, and how we can use this technique into the determination of the non-structural impurities. So mass balance operating to specifications like internal, external quality control and maintenance. It require control environment like temperature, pressure, relative humidity, and vibration, etc. Material for mass determination is not subjected to the static effect, and non-volatile, non-hygroscopic. So if you have the good practice of measurement, you can minimize the measurement uncertainty. Here are some of the books which I mention here. If you go through these books, you will get a good technique procedure, how to accurately do the gravimetry, and understanding about the buoyancy and measurement and uncertainty. So I think you must everybody aware what is the mass and weight. So mass balance, we can directly putting some of the weights and then object, and we can directly see the reading in the balance, which is called the mass balance reading. But uh, uh, if we talking about the weight, so weight is nothing but is depend on the gravity, how gravity is work on the mass. So mass is a fundamental property, but weight is not the fundamental property, and it's depend on the buoyancy of the environment. So here it is the same thing. I am not going to repeat it again. So mass and weight. So generally, mass we can take as a mass balance, but there is slight difference between mass and mass balance. But it is fit for purpose in most of the organic analysis. But if you are talking about the SI traceable, then you need to do some of the buoyancy correction factor. So then your true mass may be the greater than your mass balance, because there are uncertainty which is larger than the uncertainty of the mass balance. So this is the formula we can uh, use to calculate the SI traceable uncertainty. I think this slide is repeating. I don't know how. Uh, yes. So what is the summary? So smaller bias for mass fraction ratio of organic analyte in solution. Both biases similar in magnitude and direction effectively cancel out. If you look at the formula, uh, in organic analytes solution, both sides, because we have the similar magnitude and direction, that is why they are cancel out each other, and measurement uncertainty is different. This is again the conventional mass and true mass. So as I already told you, that the mass, true mass is maybe approximately similar to mass balance, but we need to add it buoyancy correction factor.
Incoming call from push vendor identified by.
Sorry, actually, are some disturbance in between. So gravimetry estimation of mass balance, how we can uh, estimate the uncertainty in mass balance. So measurement model approach here we have used. So mass balance is equal to mass balance I, and delta R, delta NL, delta ST, and delta TC. If we add all these factors, we'll get the uncertainty in mass balance. So nominal value provided by the balance manufacturer for repeatability value means delta R we can get from the manufacturer. Non-linearity NL we can also get from the manufacturer. Sensitivity tolerance, uh, temperature coefficient, everything we can get from the manufacturer, and if we'll put all these value in the this formula, we can get the uncertainty in mass balance. Again it is same. So I will show you that uh, this is the balance specification from the manufacturer, and if you look at here, you can get the SR value and non-linearity value, TC value, and ST value. So these value you can put here in the formula uh, here, and you can get the uncertainty in mass balance. So now we'll talk about the water content measurement by Karl-Fisher titration. So Karl-Fisher titration, I will explain in the next slide. First, I will talk about the importance of water content estimation in organic compound. So always present and also present in atmosphere. You know, water is present everywhere. It is difficult to remove by heating, not detected by most of the chromatic graphic method, whether it is TLC or HPLC or GCMS. So limiting factor for very high purity material is more than 99.9%. So Carl Fisher method is one of the best method. So Carl Fisher method is also having different kind of reaction like Carl Fisher reaction, volumetric KF titration, and colorimetric KF titration, then endpoint indication, then drift as second endpoint indication, measurement model and validation issue, one transfer and direct addition. So I think you everybody know about the Karl Fischer, which is the German petrochemist, which invention this method was published in the year of 1935. So Karl Fischer titration uh, is exactly mentioned as Karl Fischer titration, not as a Karl Fischer or dash is not actually coming in the Karl Fischer. So what is Karl-Fischer reaction? If you react with methanol, sulfur dioxide, and organic base, so we'll get a complex compound. This compound is react with uh, water and iodine, so you will get uh, the water is catched by this iodine, and this will give the direct measurement of the water. So Karl-Fischer reaction is actually pH dependent. Here you can see it is too slow at lower pH, and uh, having side chain reaction at higher pH level. So optimum range is between 5 and 7. So basic ingredient of KF reagent is iodine, sulfur dioxide, buffer we are using the imidazole, and solvent we are using the methanol. So there are two types of Karl-Fischer titration. One is volumetric Karl-Fischer titration, another is colorimetric Karl-Fischer titration. So when your water component percentage is 0.1 to 100%, then it is better to use the volumetric Karl-Fischer titration. And if your percentage is 1 ppm to 5%, then you have to use the colorimetric Karl-Fischer titration. And in colorimetric, iodine generated electrochemically in situ during the titration. So here you can see volumetric titration with one or two component reagent, water measured in milligram, and it is useful for 0.01% to 100%. When it comes to the colorimetric titration, it is within the cell or without diaphragm, water measured in the microgram, and water content is 0.001% to 5 to 10%. So in NPL, we are having both the version, volumetric titration as well as the colorimetric titration. Another is KF titrated with an oven. It is not a special titration technique and indirect from the sample introduction. So we can use oven technique with volumetric or colorimetric or in combination. So principle of the colorimetric Karl-Fischer reaction. As you know, 1 coulomb is the quantity of charge transported by the electric circuit of 1 ampere during the 1 second. So 1 C is equal to 1 ampere and 1 S. So it's a absolute method and direct measurement, no calibration is required. So produce 1 mole of a chemical using 1 electron required 96,484 coulomb. So 2 ion from the I2 which react with water. So 1 mole of water 18 gram is equivalent to 2 into 96,484 C or 10.72 coulomb per mg. So this is the reaction is held in the KFT reaction in the analyte solution. On cathode, the proton will get the electron and hydrogen gas is released, and at anode, iodine production by the oxidation of iodine, and then iodine gas produced and electron is released. So that electron is measured in the form of the concentration of water. And this is the principle of the oven technique. In oven technique, there is a colorimetric cell which is linked with the uh, sealed sample vial placed in the oven, and nitrogen is passing through this sealed vial. Uh, these are some side reaction of ketone and aldehyde. So you need to take into the consideration if your sample is having ketone or aldehyde structure in your compound, then there may be a side reaction with methanol, which are also releasing the water, and your result will get the erratic. Like here you can see the aldehyde and methanol is there is a condensation reaction and the water is released here, and that water may also interfere into your result. Similarly, aldehyde is also having the same ketone and aldehyde both having the side reaction. So if there are aldehyde or ketone in your structure, then you avoid to use this Karl-Fischer reaction, or you have to use the correction factor. think this is not required. So measurement equation of KFT on the basis of water percentage, the colorimetric or volumetric method or their related uncertainty is mentioned here. If it is water impurities in ppm level, then colorimetric technique is suitable. Is and volumetric technique is not suitable. The maximum relative uh, uncertainty here is more than 5%. And here, if it is 100%, then colorimetric technique is not suitable for this one, but volumetric is suitable. So like that, you can select your technique based on the uh, water presented in your organic compound. So we have used this Carl Fisher titrator technique when we participated in the international laboratory comparison with the BIPM. Uh, we have estimated actually folic acid for the water content. So for KFT, your water in sample is equal to uh, your water for result for water in KF titration cell and additional water transferred from the empty vial. If your empty vial having some of the trace of the water, that can also be minus from the uh, your total water. Uh, Another technique is water content measurement by thermogravimetric analysis. So thermogravimetric analysis is a technique in which mass of the substance is monitored over a time as a function of temperature. So at a controlled environment, we have to uh, monitor the substance over a period of time, and we can see the weight uh, profile of the sample. So this is the TGA we are using. If you look at the TGA of estradiol, so here you can see when we yeah, using TGA for the estradiol from 40 degrees centigrade to 250. So initial release of absorbed water at 120 degrees centigrade, around 1.05 uh, milligram per gram, there is a decrease in total weight, and then subsequent release of crystalline water above the melting point 180 degree. You can see here around 180. It is around 6.23. So total water content of estradiol by TGA is 7.3 milligram per gram. So this is the result of the water content measurement by thermogravimetric analysis for folic acid. We have participated in ILC. So we have taken initial weight 8.37128 mg. The medium was nitrogen. Operating temperature 30 to 150. Whole time was 15 minutes at 150 degrees centigrade. This temperature program was given by the BIPM, and the quality control procedure, we have used a certified reference material to calibrate our TGA instrument, and 3 experiments were done. So results are mentioned in the table. So total water means mean value we get 7.3 mg per gram. So these are the thermogravimetric analysis thermogram, first run, second run, third run, and fourth run. So these are the specification for the TGA machine which we have used, and this is the temperature program. Now comes to the case study. Uh, while you after means you calculating all the secondary impurities, uh, how purity assessment will be done. uh, Here actually I am using only water content or non sorry, volatile secondary component, because sulfated ash and some other techniques we cannot taken because time is short. So in sometimes in next lecture we'll talking about these other technique. So based on the uh, secondary component estimation, here I am discussing the trimethoprim purity assessment. Trimethoprim purity assessment we have done with the pharma affiliates. It's a pharmaceutical company which is working on the pharmaceutical impurities. So first we have to decide the acceptance criteria for a particular API. So in description we have to check is a white or yellowish white powder or odorless or almost odorless. Then number two is identification. So identification we can do by the FTIR, by mass spectra, or by proton NMR. And then LOD, loss of drying, is not, it should be not more than 1% weight by weight. And then sulfated earth, it should also not more than 0.1% weight by weight. And then chromatographic purity, which can be estimated by the HPLC. It should not be more than 99%. And then assay by mass balance method, not less than 98%. This is the criteria given in the IPC monograph. So as per the IPC monograph, we have to follow all the acceptance criteria. So what is the standard procedure, test procedure we have opted here? So description of the substance, as I already mentioned, take nearly 1 gram of the material to a petri dish and check the visually whether it's a white background or yellowish, or what is the uh, color, or it's a odorless or almost odorless. Then you have to do the identification by FTNMR. So we have to do the FTNMR with ATR technique and compare the spectrum with the trimethoprim RS reference standard, which is mentioned in the IPC monograph. So there is a fingerprint region. If your FTIR is matching with this fingerprint region, then you can say your compound is absolutely fine. Then it is identify the mass spectra. So uh, another technique is proton NMR. So proton NMR is done on the Bruker instrument, and you you can see here the probe and everything detailed. So whatever the results is coming, uh, there is, it is used for the identification of the uh, organic compound or impurities. If any impurities is present over and above the 99%, then we have to taken care of that impurities. Then loss of drying, and then sulfated ash we have to calculate. So this is the process of overall process of analysis. First the selection of the material, like what material you want to do the purity assessment. Then procurement of raw material. Then preliminary preparation of the sample, means if there are any kind of moisture, you, or there is, you want to make it in powder form, you have to do the things. And then choosing of proper containers and labels. So after sealing, then you have to see the determination of stability of the material, and then homogeneity and stability within bottle and between bottle. Then water content determination by the using of Gall-Fisher and TGA, then identification by FTIR, mass spectra, and proton NMR, then purity analysis by HPLC and LCMS technique, then loss on drying and sulfated ash, and then assay by the mass balance, and then give the specific value with the measurement uncertainty. So after doing all this process, your materials, you can check the impurities and purity component, and then you can calculate your purity percentage mass by mass by using this formula. If you minus from 100 percentage of water, percentage of residual residue analysis, and minus the percentage of impurity, multiply by HPLC purity, divide by 100, you will get the purity mass by mass. So first we have did the FTR analysis. So you can see here we are using the range 400 to 4000 to 400 centimeter inverse, and this spectra is exactly matching the spectra mentioned in the IPC monograph. So it means it is a trimethoprim, and then you can see the chromatographic value which is done by the HPLC, and we have taken 3-4 measurement, and then we'll get the average value of the chromatographic purity. And actually this is again repeating. I don't know what happened. Yeah, I will talk about the chromatographic purity. So determination of by liquid chromatography. Here we use the liquid chromatography. So test solution, dissolve 25 gram of the substance under the examinate in the mobile phase, and dilute 25 ml with the mobile phase. Reference solution standard, we have taken 1 ml of the test solution of 10 ml with the mobile phase. Reference solution B, we have used the dilute 1 ml of the reference solution to 20 ml with mobile phase. Chromatographic system we mention here, and this is the name of the impurity we observed. These are the relative retention time, and this is the correction factors. Like that we get the chromatographic purity as well as the impurity, and this is the calculation we have done for the mass balance biases. So S is percentage mass by mass equal to 100 minus percentage of loss of dyeing, percentage of sulfated ash, and minus percentage of organic impurity into HPLC purity divided by 100. So you can see here the final results on the basis of all the analysis we have done, like FT and MIR complies. The IR aspect of the sample should be incorporated, is matching with the spectrum of the trimethoprim as mentioned in the RS, and loss of drying is 0.3%, and sulfated ash is 0.0%. Chromatographic purity is 99.9, 99.7, and 99.7. So mean value is coming around 99.8%. So property value is coming 99.46. So another important factor here is uh, measurement uncertainty. So at every step there are two type of measurement uncertainty, type A and type B. So type A is nothing but it's a uh, if you repeating your same measurement more than 2-3 times, you can get the type A error, and type B error maybe in terms of the several factors. So I'm not going into detail of the type B because it will take actually in so here final value is 99.31% with measurement uncertainty 0.19%. This is the specific value. Now it comes to the certified reference material, because I am working in the department of certified reference material, which is termed as the Bharati Nirdesak Dravya. So it's my sincere duty to introduce you about the certified reference material. In India, we are actually buying 10,000 crore CRM from the uh, overseas country every year, and huge amount of the revenue is going outside. So to cater the need of the nation and industry, we have started this program which is called Bharati Nirdesak Dravya BND. And so far we have prepared in association with the reference material producer more than 150 BNDs, which is already in the market in the area of the cement, in the area of petroleum, in the area of polymer. Some of the BND will show you. So how we can make the BNDs. So first I will talk about the reference material. So reference material is a material which is characterized by a metrological valid procedure for one or more specified properties, accompanied by a certificate that provides the value of a specific property, which is associated its uncertainty, and a statement of meteorological traceability. And this is the procedure of making certified reference material as per the ISO 17034 2016 requirement for the competence of the reference material producer. So these are the steps which is I already mentioned in another slide. So like processing of material, then sample preparation, then homogeneity, stability, and then we have to value assignment, and finally certification of the CRMs. So these are the list of the RMP which we with which we are working right now, like BPCL Mumbai, ASVI Technology Ahmedabad, HPCL Visakhapatnam, Global PT New Delhi, SUMS Technology, and CSIR IITR Lucknow, Jalan Company Delhi, Mintum NCCBM Ballabgarh, and Simphar Dhanbad. So number of CRM and associated RMP country-wise. So if you look at the Japan, it's having good number of CRMs as well as number of reference material producer RMP. But if you look at the India, we have only few number of CRMs, and sorry, it's moving actually slide, and total 12 RMP right now. So CRM is the essential requirement of each and one, because it's having very costly, and also there is a validity of the CRM. So if we start indigenously producing the CRM, it can save a lot of revenue as well as time. So this is the major test facility we are having at the CSR NPL, like HPLC, APR, ICPMS, ICPOES, QNMR recently we have installed, LCMS-MS. This is the in-house BND, SAM Caliere BND, which is used for the calibration of the SEM. this is the BND alpha aluminum for the calibration of the XRD. So this is another BND. This is polystyrene BND used to calibrate the FTAR. It is developed by the NPL scientist, which is very cheaper, and it is around uh, 200 times cheaper from the BND or certified reference material available in the market. So that is all from my side. Thank you very much once again, and I hope whatever I have tried to convey here, you will definitely get some of the flavor. If something you want to ask me, you can ask me through the chat box, or I will share my email ID to you. I will try to answer your queries. Thank you very much, and thank you so much. Hello.
Thank you very much, sir. Thank you for your brief and very interesting presentation. I would like to request you to kindly unshare your presentation, please.
Pardon?
Sir, kindly unshare your presentation.
Hello, sir. Your voice is not coming.
Thank you. Thank you very much. Thank you. Thank you. I would like to request all the participants to kindly switch on their videos so that we can take few photographs. Atulji. For the social media platform. And my request will be kindly join our social media platforms so that we can also be motivated to do better. We are taking regularly